Okay, welcome everybody. Happy Monday. Y'all had a good weekend. We have just 2 or 3 slides left in chapter 13, and then we're going to move over into 14 and think about antimicrobial agents, some details about that. And I think this slide is really a perfect one to kind of start thinking about that. Although we're looking at like different potential chemical agents that are possibly effective against specific microbes. We can also think about antibiotics in a similar situation where we could create a lawn of growth. We could apply antimicrobial discs, antibiotic discs, onto the plate, and then after we allow it to incubate for a day or 2, we can measure the zones of inhibition and ultimately get some insights into specific drugs of choice. Lots of times when we think about trying to identify specific infectious agents, one of the things, hopefully one of the things on our radar would be, how are we going to kill these suckers? If you're truly trying to combat these infectious agents, cultivating the organism, growing it up, and then testing against specific types of antimicrobial agents helps you to choose the specific agent that will be the most effective. That's not always what happens when we think about trying to treat infections. Sometimes you go in and they kind of just use their instincts. They go with their gut. And they're like, this is what it most likely is. They don't grow the organism up. They don't do any sort of antimicrobial testing. They prescribe you with the drug that they hope is going to be effective. Maybe it is, maybe it's not, all right? Lots of times, those drugs would be under the umbrella of a broad spectrum, and we're going to talk about that in the next chapter, all right? So, with that, as we're looking at these 2 plates, I want to shift away from thinking about different chemicals like, you know, we talked about all sorts of different things. Peroxide or bleach or other things could be applied to these discs and possibly seeing some inhibition. If we think about the antimicrobial antibiotics, these antimicrobial agents, it's a similar idea. And in these plates, we'll describe here in a bit, this disc diffusion or Kirby-Bauer technique uses some big plates, spreading the bacteria on, and then allowing the antibiotics to possibly do their job. So we measure the zones of inhibition, basically the diameter of those circles, and typically the biggest zone would be the drug of choice. There are definitely some other considerations when we think about this stuff. We need to look at the patient histories, find out things like maybe some allergies. If the patient's allergic to penicillin, you're not going to prescribe penicillin, even if it looks like it's the best drug, right? So there are some things we need to take into account. There's also other things as we look that you might see some zones that look pretty comparable, all right? And that opens up avenues to think about possibilities of utilizing something maybe together. A cocktail approach where you use a couple of different antimicrobial agents with the hopes, and we'll see this in the next chapter, there's multiple ways that these drugs can target a bacterium. And if you pick 2 different classes of antibiotics that focus at different mechanisms of the pathogen, you try to get it from any angle you can, right? And hopefully that also helps to minimize some of the possibilities with resistance, right? So we'll see some more of that as we progress. So we're still thinking about chemical agents, and in this case, some disinfectant monitoring how good it is at killing stuff. We can also think about trying to inhibit the antimicrobial agent to show that stuff could grow. And that's kind of what we're looking at here with this. So you put some of this on there, incubate it up, and look for colonies. Is there potentially something in that disinfectant that's able to survive or not? You would hope that cleaning agent, whatever it might be, doesn't allow microbes to grow within there. That'd be an issue. So this is the inactivator that you're putting it into and seeing what happens. And I think we're right at the tail end. So this CRE is a type of antimicrobial carbapenem resistant enterococcus or enterobacter is issue. Nosocomial infections are hospital acquired infections. And we might think about something where possibly the medical device sets the stage for bringing organisms from one patient to the next. And that seems pretty disturbing and disgusting. As we think about some of these things like this duodenoscope, the potential to get it thoroughly sterile is pretty much not there, right? We try to cleanse it, we try to remove, but the issue is still there. And we can see infections in this situation being spread from one individual to the next. And some of those, like this, that are highly resistant. Okay, so that's end of that. I gotta stop share and reshare. Okay. All right, so 13, chapter 13, really the end of that, I feel like is a pretty good segue into thinking about the antimicrobial drugs. There are a plethora of different agents that we might think about as being possibilities, all right? Initially, the first antibiotic was penicillin, and it was mass produced in the early 1940s. It was discovered about a decade before that. And we'll go through some details with that. So upon its, well, discovery and really the ramping it up to widespread usage, the thought was, we have just found this maybe magic bullet. We found this drug that seems to be the cure-all. We can use this to treat anything and everything. And I think you recognize now, I hope you recognize now, that is a huge mistake, right? It's definitely effective at killing some stuff, and definitely overused and misused, lending to issues of resistance, right? Antimicrobial resistance that we will talk about, and it should definitely be on our radar. It's not something that is new, but we definitely think of it as becoming a bigger problem, right? Bacteria and fungi have had capabilities of producing some of these substances that we think of using to treat specific infections. They've been doing this in the environment for a super long time, just trying to survive and keep others away. And in turn, the others that are trying to be kept away have started to develop strategies to deal with some of these substances. Resistance has been there, all right? It's just a matter of us using these agents and having some of those potential pathogens or true pathogens that are there with some of these capabilities to resist. And then it's basically like artificial selection, where we are the driving force of that. By bathing in antimicrobial agents, we set the stage for those that do have resistance to truly proliferate, all right? So resistance has been there, but we're a huge amplifier of that as we think about things like this, all right? And we will go through more of that later. So there's definitely all sorts of things like this from, World War II touting the benefits of penicillin. They really say, a lot of the idea with this and the discovery and use of it truly helped the Allies with the war. All right. There are definitely bonuses with this, but then we also can think right in this window of time. All right. When penicillin first was utilized, we could see it being pretty effective against something like Staph aureus. All right. And over the course of just a handful of years, we see some staphylococci that no longer are targeted by penicillin. And we see resistance. Like I said, that stuff, technically, we've already have some of these within the population that have that. So we focus in on it. And then we get lots of strains, lots of individuals that seem to harbor these resistant forms. This is an issue, all right? And shortly after the widespread use, we start seeing some of these problems. And it's still something that plagues us today. There's all sorts, and I wish I could just look at this and start spouting off all of the things. I don't know. And here's a card that probably does spout all of that off, but you can't see it. All sorts of different plants, roots, herbs, all sorts of things that have some potential properties to treat numerous types of ailments. Some of these things could be highly effective and some of them potentially not, right? It really kind of depends on the circumstances and what you're truly trying to deal with here, right? With that said, lots of these things, as we think going back, centuries or thousands of years, looking at this and all of the possibilities with these different types of plants, we think about potentially tapping into some of this. Bioprospecting is a situation where we look at different chemicals produced by plants, typically, but it can be other things as well, that we might be able to utilize. We recognize this root or whatever has some specific properties. Well, what chemicals within that truly are initiating that, and can we deal with that in a laboratory situation? Lots of antibiotics. We think of some natural occurring situation, some organism producing it, and then in a laboratory situation, we may potentially modify it. Biochemistry, manipulating those molecules and creating some sort of hybrid or a full-on synthetic antimicrobial agent. So there are numerous things that we may be thinking about as well with something like this, right? And with issues of things like antibiotic resistance, tapping back into some of these historical or ancient texts to look at some of the mechanisms that they may have used, we may bring some of that stuff back and recognize these things could be highly effective at treating something that our antibiotics no longer are, right? And I think that is pretty interesting. So this microbiologist, this scientist, Paul Ehrlich, was influential in the discovery of this compound, 606. It makes me think of like, WD-40 or 409, right? All of these different things that are multiple attempts. And finally, on the 606th attempt, we found something that worked. That's perseverance right there, right? So this agent that he discovers proved to be effective at treating syphilis. So spirochete, organism that, well, we've seen some images of that. And ultimately thinking about treatments for sexually transmitted infections, huge, right? As we think about this penicillin, we could back it up. Another one, right? A quick shot of that and forget about it, right? Maybe. So for our penicillin, for our antibiotic, Alexander Fleming was the first to discover penicillin. in 1928. And it was, from the story I recall, it was kind of an accident when this was discovered. So back in the day, doing microbiology, little bit different than we think about today. A lot of the Petri dishes, and you can see some of them on the bench top. These Petri dishes were glass with loose fitting lids, glass lids that would go on the top. I described before all of the, like the sleeve of plastic Petri dishes that we get, that sleeve is sterile, and they sterilize it with some ethylene oxide, some gas that helps to kill anything within there. The glass Petri dishes, that's not the same idea, right? And in turn, you could make your media, you could put it into those, and you could cook those up, and it would be sterile. Or you could cook the dishes first, sterilizing, and then adding your media aseptically. Similar idea, but it's glass and it can be reused, which is kind of a bonus. There are some thinking, you know, microlabs, we should use just glass Petri dishes. Wouldn't that be great? And the people in the stock group say, hell no, I don't think so. With hundreds of plates being made every week, that's a lot of stuff, right? It's a lot of issues. Test tubes are a big enough problem, right? So all of these glass dishes, I said the lid really pretty loose fitting, all right? That sets the stage for some potential issues, maybe some contamination, right? So he's looking at Staphylococcus aureus is what he's got plated up. He's dealing with these bacteria, not thinking about trying to find something to kill them, right? That's what happened by accident, all right? So he comes into the lab the following day and notices, or maybe 2 days later, whatever, one of the Petri dishes that has the Staph aureus, the lid was sort of off, right? Exposing part of that plate just to the air. And we talked before, bacteria all over the place, right? So are fungus. And fungal spores floating around have the potential to settle down on a plate and start to grow. So then all of a sudden this mold pops up. That's what he sees. The plate's opened a little bit. There's this blob of mold growing on the plate. And his first thought is, that experiment is ruined. I'm gonna have to set it up again, which I'm sure he did set up the experiment again. But as he looks at the plate, he recognizes where this big fuzzy mold is, penicillium, where this mold is growing, Staphylococcus is not growing. And that makes him wonder, what is it on this plate within this mold that's inhibiting the growth of this bacterium? And it's kind of back to the picture of those plates where you see those zones, that area where nothing's growing. He sees that stuff and recognizes there's something going on in this fungus that's inhibiting the bacterial growth, all right? So he recognizes it, discovers penicillin, the substance within the penicillium mold that is the antimicrobial agent. Flory and Chain, a couple years later, a handful of years later, basically ramped up the production. So growing these things, not just on a little Petri dish, but in a gigantic bioreactor where they've got lots of nutrients pumped in there and the mold is just thriving. They're able to grow up mass quantities of the mold and extract the drug, the penicillin, out of the fungus, right? So they ramped up the production and then were able to purify and test it as an agent in animals and humans in the very early 40s. And as I said, it explodes from there, all right? So the discovery, the industrialization of it truly as we think about ramping it up into widespread usage. Waxman, Selma Waxman, and there is a Waxman Foundation that's still doing massive research with antimicrobials and other microbiology. But he was looking at these actinomycetes, these different, so not fungus, but bacteria, but similar idea. So some types of fungi produce antimicrobials, some types of bacteria will produce antimicrobials. Not everybody, all right? And the actinomycetes are pretty good at dealing with this. So they are a type of soil microbe. There's a lot of different actinomycetes. And some of these, like streptomyces, a type of actinomycetes, is a soil microbe, and it ultimately has the capability of producing some pretty good antimicrobial agents. And there's a handful of others, all right? They also, some of these, like the streptomyces, helps to impart the odor of soil in the first place. I think that's kind of cool. And there are lots of variations with this. So I recall a class, it was an industrial microbiology class where we were looking at all sorts of different things. And one of the one part of that lab, the focus was to try to isolate a microbe that was effective at killing other microbes. We wanted to find somebody producing an antibiotic. In industry, when we're trying to find antimicrobial agents, what we were doing in the lab was pretty similar. You find a soil sample. In this case, it was some compost from the professor's yard. He brought in these soil samples, and we took little bits of it, tried to plate it out on some Petri dishes that didn't have lots of nutrients in there, because environmental organisms, although compost is probably very rich in organic material, just in the environment, not huge amounts of nutrients. So multiple rounds of trying to get bacteria that are actinomycetes and not just other bacteria, get them all separated out. And the actinomycetes started to grow on these plates. And you've all seen some things on the Petri dishes, maybe some environmental things that looked very odd. The actinomycetes have some crazy looking colonies, bright colors, kind of liquid, almost little droplets of substance that they are secreting, potentially antimicrobial properties, right? Really pretty interesting. And unlike any types of cultures that I'd ever seen, really pretty impressive just to see some of the different pigments, red and blue pigments that some of them were producing. Really kind of crazy. So that's the same, similar idea like the discovery of penicillin and trying to ramp it up. That's kind of what's happening in this situation, but it's not fungus, it's bacteria. And trying to find all sorts of different organisms and then test them against specific agents like those Petri dishes with those big zones. You could have numerous types of bacteria that you've created a lawn of growth on, and then these agents soaked in a little disc or dropped just a drop of it onto the plate to see, is there a zone where we've killed these organisms or not? And able to isolate and generate numerous types of agents. So I mentioned broad spectrum as we were wrapping up the last chapter. This shows some of these details, some of these ideas as far as what's happening here and how stuff like resistance or super infections can manifest. So in this case, we've got a couple of different frames that we want to think about what's happening. This patient's got a handful of organisms in their system that we would say are the normal ones, the normal flora. And the little guy right here that's the pathogen, this is an opportunistic pathogen, so it has the opportunity, given the proper circumstances, to become an issue, right? This pathogen, we start treating with some broad spectrum antibiotics. And in this case, fortunately for the pathogen, or unfortunately for us, the drug that was prescribed is very effective at killing the good guys, but it's not effective at killing this bad guy. And it may harbor some resistance within it. It could just be that the specific class of agent isn't effective at targeting that, but it's likely it's got some sort of mechanism to allow it to resist that agent, all right? As we've wiped out the bulk of our normal flora, these guys are left over, and now they've got ample real estate. They've got plenty of room to proliferate, lots of food. And as this happens, they become the dominant population. You could think about something like this with Clostridium difficile. And you could have all sorts of good guys in your gut, and then you've got a few of these bad guys, all right? They're there, they're held in check by the other ones, but when you take the broad spectrum drug, it opens up the window for this to occur, all right? And oftentimes in those situations, you're not taking this broad spectrum drug for some sort of GI issue. It's something completely different, all right? But as it turns out, it wipes out the GI bacteria as well, all right? Opening up this can of worms, if you will. It sets the stage for them to proliferate. You hope that if you go in and they are truly trying to figure out how we're going to treat this, that there is some cultivation that happens, that they actually are able to say, this is the drug we should use, and not something that's just a kill them all sort of approach with this broad spectrum agent. That's what you hope. And that might minimize this, so it can't take over and cause a super infection. So it truly depends on the circumstances. It depends on the type of opportunistic or true pathogen that could be in there. What do they bring to the table? And the specifics with the types of drug. There, as we look at this, we're looking at the effectiveness at a couple of different ways that we may take these drugs in. So oral, intramuscular, or intravenous administration of the antibiotic. Lots of times we think of taking the pills, right? Oral administration. It takes time for these things to do their job, all right? So we've got arbitrary time on the x-axis, handful of days, potentially, all right? And on the y-axis is the plasma concentration. So the amount of the drug in your circulation, all right, over the course of time. With the IV administration, it's the bottom of our list, but it's the one that is the quickest to be detected, and it would make sense. If you are injecting this into your circulation, you would expect almost immediate delivery of that agent, right? So shortly after it's injected in, we can see a spike with a very high level or a high titer, the amount of that agent within the circulation. And then it starts to decrease. You've had one shot, right? It's not something that we're continuing. So you would expect to see it kind of spike and then taper off. The intramuscular, similar idea, an injection, but it's not into your circulation, it's into the muscle. So that in turn takes longer as it needs to diffuse and then make its way throughout the body. So we can see it not spiking as much, but at time zero, it is a higher amount that's actually in your system. A day, because if we're giving these time in days, at about a day into it, we've got a pretty high amount of it, and it starts to decline as well. The oral administration, this takes longer after the first dosage with this, takes longer for it to get digested and ultimately make its way to be detectable within your body. About the same as the intramuscular, it's a little lower, but at about a day or so, we've got our peak where we could detect that within our body. And then it starts to wane as well, all right? This is if we're, think of just a dosage, right? One dose of this in any circumstance. If we're thinking about the oral administration, oftentimes we think about repeated dosages. That helps to bring this concentration up and get it potentially close to up above if we keep taking this every day or twice a day, right? For 2 weeks, right? Bringing those levels up. And the same situations could be happening with something like the IV administration. So it does depend, right? And it takes time, considerably more time, really, if we're making a comparison to the IV versus oral or intramuscular a day before we see some of these peaks versus maybe a few hours, right? So it does depend. And like I said, most that we think about, it's definitely easier if it's some pills that you have to take. Whether you comply with that is a different story, right? This image, I'm kind of torn by this. This is parental expectations of antibiotics over the course of a decade, little over a decade, from 2000, and then resurveyed in 2013. And then trends, what do they think would happen or should happen if the doctor does not prescribe one, and I think that one is needed, I'm going to go to another doctor, or if I expect that antibiotic, am I less satisfied if I don't get it, or I'd rather give my kid an antibiotic that's not needed than rather just, waiting to see, do they recover? Couple of different questions with this. And we've got surveyed Medicaid versus commercial insurance. And as I'm looking at this, I'm like, well, this isn't that far back, right? These issues, I feel like, are problems. I'm like, I can't wrap my head around it. Yes.
Isn't it bad to take antibiotics that you don't need because you know what you need?
Heck yeah, it's bad. So that there's numerous things on here that just kind of frustrate me. I'm like, I don't care which one we're looking at, blue or red, matters not to me. There are issues. And taking stuff, and back in the day, little kids, when I was a little kid, you'd get a cold. If your mom took you to the doctor, you could almost guarantee you're leaving with antibiotics. Almost a guarantee. Are they going to be effective? Nope. But mom feels good because she took you in and she left with some amoxicillin. Tastes delicious, but it's not going to help you. So, question?
Not really, just more of a comment. So the difference between those two, I say this from a position, my first child was born mid-90s, and I was on when he was born.
Yeah.
I couldn't afford to not be at work.
Yeah.
I mean, that was me, like, if I went to the doctor, he had something going on, his was ear infections, he got them a lot, and I didn't get an antibiotic, I would go somewhere else. Because that was the only way I was getting back to work.
Right. And you want that.
Now,
and,
it didn't help him in the end.
Lots of, ear infections, lots of times it's bacterial. So we're not talking about going in with a cold and getting antibiotics.
Right.
It's an ear infection typically is bacterial. But as you said, it didn't really help them because they repeat, they create, lots of the bacteria can create some biofilms, and that sets the stage for big problems, for it not really being able to treat it like it should. Pseudomonas, that one we showed that looked kind of green, very resistant, and it's a good one with ear infections. So, yeah.
So in a situation like that, where like if they were giving antibiotics in the past when like they didn't need them, how does that like affect people when they're older to that medication?
So that's a good question. So we could think about something like that. maybe if I flip it back to this. And if you, it doesn't matter where, where we're talking in the body, if you're prescribed some of these antibiotics, you got a cold, a virus, and you prescribe some antibiotics, the potential for that antibiotic to kill other things, well, in this case, that's gonna happen. It's not killing the pathogen because the pathogen's a virus, and an antibiotic isn't gonna kill that virus. How do you kill something that's not alive? So what happens, what could potentially happen with the misuse or the, over prescription, or, you're prescribed something and you shouldn't be, right? You could set up a situation where some of the bad guys are the ones that become the dominant ones because we've killed off some of these good ones. And so you can create a situation in your own body where you've got some organisms that we would classify as being resistant. Do they cause issues? Maybe, maybe not, right? Staph aureus on our skin is all fine and good, but if it gets through the skin, then it can be a potential pathogen. And all of us, we could swab our bodies and we could find Staph aureus. And if we were to test our Staph aureus, each of our Staph aureus, with a whole spectrum of antibiotics, we would see variation. And some of us are walking around with some drug-resistant forms of Staph aureus, and others probably have some that penicillin could treat. It just kind of depends on the individual. It also depends on where you swab, right? If you've got MRSA, if you've got methicillin-resistant Staph aureus, it's not completely covering your body from the tip of your toes to the top of your head. It might be right on the side of your nose, and that's where the MRSA is. And over here is normal Staph aureus, right? It truly depends on the circumstance. And if where it was is the one that got in, and you see that infection, it could be the bit on my arm and I scraped it, and now I'm infected with staph, but it's not the drug-resistant one that I've got on my face, right? Truly depends. It depends on what you were treated with. It depends on, as a little kid, did I get lots of colds and every time I was dosed with amoxicillin? Maybe. And what did that do? That's hard to say, right? But later in life, we do think of some of these potentials where your flora is maybe not the same as what you started with, and maybe some of it is a resistant form. And it's things like that that sets the stage. And this, I definitely understand that, your kid needs to go, gotta go to daycare, gotta go to school, one way or the other. Those things are important. But issues of like a cold and this, it just gets me. And I'm like, I don't care which color this thing is, those are problems. But I do understand those issues, right? It's some of these that I'm just like, that's odd. The trend we are seeing, and here both kind of an increase from 2000 to 2013. I'm not going to be as happy, and it doesn't matter, right? And yeah, interesting. Okay, I mentioned all sorts of different classes of antibiotics, right? There's maybe a couple of approaches we might take in this kind of cocktail situation where we use a couple of different agents, maybe hoping that we can target from a couple of angles. There are lots of drugs that fit into these categories, right? I don't want to get bogged down and talk about all of the specific ones. I don't expect that you are going to know all of the names of these drugs. I don't think that truly is important in here, right? If you are in a clinical situation, having a pretty good handle on these would probably be to your benefit, all right? But for our purposes in here, the actual names of these drugs, I don't want to get bogged down with. With that said, there's probably a question or two on there on the exam that's going to say, you know, something maybe about penicillin, but really it's cell wall inhibitors that I'd like to focus on, right? You may see a few of these, but I'm going to try to select questions that aren't specifically talking about, you know, whatever of these, you know, bacitracin or whoever, right? It's the bold stuff, right? Cell wall, DNA or RNA synthesis, the plasma membrane, ribosomes, or metabolic pathways. These are the angles that we may drill those bacteria with, right? The specific drugs, yes, they are important, but I don't want you to have to try to commit all of these things to memory. I don't think that is truly important for you. Yes, sir.
The mycolic acid inhibitors, those are specifically for mycobacteria, right?
Yes. So the mycolic acid inhibitors, mycobacteria like your tuberculosis, leprosy. There are some others that have some mycolic acid. I think nocardia, but really like tuberculosis, we think about with that. So that's a good one. This, as we're on this, the metabolic pathways, there's a handful of things that fit within this category. So blocking folic acid synthesis, that's a great one for the bacteria. We don't synthesize folic acid. We get it from our diet. And in turn, the bacteria, they can. And if we can drive a wedge in their synthesis of folic acid, it doesn't hurt you at all, but it hurts the bacteria, right? And that a couple of others with these pathways, similar idea, where it's directly targeting a function within that bacteria and really having no effect on us at all. Some of the others may potentially have some effects. If we look at the DNA or RNA synthesis, these drugs can cause issues, side effects in us as well. Same as we target ribosomes, right? So there's definitely issues as we think about some of these things, all right, as far as the effect on a person. I mentioned, you know, playing with the soil and finding some sort of microbe, whatever it could be, some sort of microbe that's effective at treating a type of bacteria. There are all sorts of things out there that could be really effective, but their level of toxicity is huge. And in turn, using that in or on your body would kill you as well, all right? You don't want to use that, right? But I killed the pathogen, right? Killed the patient too, but no more pathogen, right? That's not a good thing, right? So all of these, as we're looking at it, there could be some potential issues, but we're hoping that it's more of an issue for the pathogen than it is for us, all right? And something like folic acid is, you know, it's good for us and bad for these guys, right? The DNA synthesis, so if we can inhibit cellular replication, or we can inhibit the fact that these guys can't make their proteins, they can't build the materials they need to replicate one way or the other, we're going to dramatically impact their growth. Same idea as we look at the ribosomes and the machinery needed to deal with the proteins, but in this case, it's already synthesized the messenger RNA. Now we just make it so you can't build it, right? That, as we look at impacting the ribosomes, we can see different subunits that they're targeting. Bacteria and eukaryotic ribosomes are different. So for the most part, we think, well, this is a good thing, but we should put the brakes on for a second and go back to whatever chapter it was when we started talking a little bit about eukaryotic cells. Our own cells are full of mitochondria, and mitochondria have bacterial ribosomes. So what happens to us if we're taking drugs that are impacting the ribosomes of the bacteria? What do you think that does to you? Sometimes you take these drugs and it wipes you out, right? It makes you super tired, right? Lethargic. I would think something that's inhibiting or blocking the ribosomes is going to target your mitochondria. And you felt crappy, and now you feel even crappier because the drug is wiping you out, right? That's probably, that's my guess, as far as something that could be happening here. The plasma membrane, this also an issue because our cells also have plasma membranes, but this gets in and basically inhibits some of the production of the cell wall or plasma membrane, excuse me, cell wall right here. With that, the cell wall, now we've got to step up because our cells don't have cell walls. So targeting the bacteria this way, this is a good class of drugs. The penicillin that we started the chapter with fits within this category. We say these are beta-lactam drugs. The beta-lactam is a big ring molecule, penicillin, and its cousins, if you will, fit within that category. And there are a few others. We can see vancomycin down below. I mentioned the methicillin-resistant Staph aureus, or MRSA, fitting up within this category. And then issues with it, if we do find a patient that's dealing with this, the next step would be to treat with vancomycin. Vancomycin is a pretty potent antimicrobial agent. I believe it requires some IV administration, if my memory serves me. And in this case, you would hope that it's effective at treating it, all right? I remember back in the day, things like the MRSA, the methicillin-resistant staph infections, pretty stinking common, all right? MRSA, happening, but not super common, something that we're a little concerned about, but it's fine. And then all of a sudden, it's a huge issue. It's not fine. These are big problems. Well, we treat those people with vancomycin. Don't worry, we can kill that staph. Don't worry about it. All of a sudden, we recognize there's some vancomycin-resistant staph aureus. Don't worry about it. It's just a little, right? We're fine, right? Now we see it as becoming, it's a bigger problem. And we don't really, I mean, we definitely still talk about MRSA, but we also talk about VRSA, vancomycin-resistant Staph aureus. And at that point, then we throw our hands up and we're like, well, I don't know what I can do for you, dude, right? How do I treat you when the drugs that we've got on reserve, now there's resistance, right? So definitely some potential issues and concerns. So multiple ways that we may try to combat. And again, thinking kind of the bold terms, these are how we really want to think about the classes of antibiotics. So with that said, there are, and here we're thinking the cell wall inhibitors, handfuls of drug names. I don't care that you know any of this stuff, but you can see some derivatives, some modifications, some biochemistry where we can manipulate these molecules a little bit. Penicillin from the fungus Penicillium, this is the beta-lactam ring, right? And then we can see some alterations with it. They still have these rings, right? Still has the beta-lactam ring, but the R group, the functional bit on this, slightly different or modified, all right? And a different type of drug and different type of activity that we might see, all right? Penicillin as a cell wall inhibitor, the gram positives with those thick cell walls, that's what we think about as being more likely to target, all right? Inhibiting the synthesis of that penicillin, pretty stinking good against gram positives, a few gram negatives, all right? So this, couple the ways we administer down below. Penicillin V, similar over here with our gram positives, slightly modified with an oxygen in there. Ampicillin, amoxicillin that we mentioned a second ago, and then methicillin, the MRSA, right? And when we say the staph is, methicillin resistant, it's more than really just that. We start looking back at these others, and it's likely resistant to penicillin and amoxicillin as well. So with that, the resistance with some of these, some organisms have an enzyme called beta-lactamase. If it ends in A's, you know it's an enzyme, right? Beta-lactamase is an enzyme that sees this ring and says, delicious, I will eat that. They destroy it, they split it in half, and now what should be an active molecule has been destroyed. Penicillin's not effective against organisms that have beta-lactamase. They take penicillin in or its derivatives and destroy it, right? And that's one mechanism that we can resist. All right, guys, I will stop with that. Farewell. I hope you have a good day. Go finish your course of antibiotics that you've saved. Oh, that's terrible advice. I'm just kidding.
Or don't.